Are you keeping up with the latest in non-opioid research? Missing critical preclinical insights can set your work back significantly. Today, we're diving deep into GsMTx4 derived peptides, an intriguing strategy emerging for selective mechanical pain modulation. These compounds offer a unique research pathway, distinct from traditional approaches, and hold substantial promise for future studies into managing chronic discomfort. Let's explore their origins and current research findings. GsMTx4 is a 34 amino acid peptide, originally isolated from tarantula venom. An intriguing source, early research identified it as a potent modulator of mechanosensitive ion channels for study. This distinction explains why GsMTx4-derived peptides show high selectivity for mechanical pain research. Researchers shortened the parent molecule to improve drug likeness for further investigation. Peptides such as Pept01, Pept02, Pept03, and Pept04 were developed. Each represents a rational attempt to isolate pharmacologically active regions for study. These peptides fall into two structural categories, critical for understanding their action. Type 1 peptides derive from loop regions 2 and 3 of GsMTx4. Type 2 peptides primarily originate from loop 2, a key structural refinement. This refinement directly informs molecular motifs responsible for channel modulation. Compelling data comes from well-established animal models of mechanical pain research. Studies include carrageenan-induced inflammatory pain and chronic constriction injury models. Systemic administration of these peptides significantly reduced mechanical hyperalgesia in studies. Doses around 270 micrograms per kilogram produced anti-hyperalgesic effects. These effects were comparable to morphine within experimental models for mechanical readouts. Unlike opioids, these peptides did not reduce thermal or cold hyperalgesia, highlighting selectivity. This points to a highly selective mechanism of action for research exploration. The selectivity of these peptides for mechanical pain is truly a fascinating area of study. This directed approach could redefine our understanding of pain pathways. If you're finding this deep dive valuable, make sure you're following Peptides.today for all the latest in peptide science, and connect with me, Sonia Rao, for detailed clinical cadence insights. We're consistently tracking what's next in the research pipeline, ensuring you stay ahead of developments. Next, we'll explore key targets and future research challenges. Mechanistic studies strongly implicate TRPV4 as a primary mediator of these effects. TRPV4 is a mechanosensitive ion channel involved in sensing mechanical stress in cells. In mice lacking TRPV4, the analgesic effect of Pept03 was largely abolished. This finding provides strong evidence that TRPV4 plays a central role. In vitro experiments showed Pept01 and Pept03 inhibited channel activity. Pept03 consistently demonstrated the highest potency among tested compounds in labs. Its N-terminal tryptophan enhances interaction with the lipid channel interface for studies. One encouraging aspect is what these peptides do not do in research. Preclinical testing shows these peptides did not induce tolerance over repeated dosing cycles. They also failed to produce conditioned place preference, indicating no abuse potential. This lack of reward-associated behavior aligns with their non-opioid research mechanism. GsMTx4-derived peptides are still at an early stage of preclinical development. Extensive toxicology testing, pharmacokinetic profiling, and formulation work are required. Peptide drugs face challenges like poor oral bioavailability and limited systemic stability. Alternative delivery methods like subcutaneous or transdermal systems will need exploration. Unregulated online vendors sometimes sell so-called research peptides directly. These compounds are not verified, and their purity and safety are entirely unverified. The data on GsMTx4-derived peptides position them as a scientifically grounded approach to selective mechanical pain research. Their unique mechanism avoiding opioid pathways makes them especially compelling for further study. While the road ahead requires substantial investment and development, these peptides represent a hopeful experimental step. Stay ahead of the clinical curve. Follow Peptides.today and me, Sonia Rao, for cutting-edge pipeline insights. Before we sign off, a quick legal disclaimer. This podcast is for informational purposes only and is not medical advice. All compounds discussed are for research use only and not for human consumption. Always consult a licensed healthcare provider for medical needs and treatment.